a prokaryote? So just now I explained pro. The word pro stands for the before or primitive one, and karyon always stands for nucleus. Okay, so the primitive nucleus is the nucleoid. So the word you have to remember whenever you come across with prokaryote, nucleoid is the one. What is nucleoid? Nucleoid is nothing but the chromosome material, all the genetic material which is not surrounded by nuclear membrane and is dispersed in cytoplasm. See, in the cytoplasm it is dispersed, the coil structure. It's the coil structure which you find only in prokaryote is nucleoid. Now when you take eukaryote, U stands for true or the advanced one. So advanced nucleus means the nucleus is bounded by a nuclear membrane. So it will be in continuation with 3 layers. All the notes related to whole structure of nucleus we will discuss in the next sessions. Okay. Then here, when you take the definition wise, this is the basic difference between prokaryotes and eukaryotes. The remaining points are very simple one. Which organelles the prokaryotes will have and which organelles eukaryotes will have. The basic difference. Okay. Then after pro and eukaryotes, if you take the structural differences. So here they used a word in prokaryote, tough cell wall. Understand the word? Tough cell wall. They didn't use cell membrane. They used the word cell wall. When you take eukaryote, if it is a plant cell, you will have cell wall. If it is an animal cell, you won't have cell wall. But plasma membrane is present both in plant and animal cells. Don't get confused at that point. Okay? Eukaryotic cells, if they are plant cells, they will have cell wall and plasma membrane. Both the membranes will be there. But if eukaryotic cell is an animal cell, they will have only, the word only indicates they will not have the cell wall. The animal cell will not have cell wall. That's the basic difference. Then they does not have nucleus, the word, the same one. Then in eukaryotes, they'll have prominent nucleus. Then the cell-based several organelles, this confusion in giving the difference. Actually, this point has to come here and this point has to go there. Cell organelles are absent. Basic cell organelles like mitochondria, Golgi apparatus are absent in prokaryotes, not in eukaryotes. Okay. So the third point, this point has to be there in eukaryotic cell column, and this has to be there in prokaryotic cell column. When you take the DNA structure is circular, okay, in prokaryote, whereas DNA structure is thread-like in eukaryotes. That's the structural difference. That's what the heading. Basic structural differences I'm discussing. Okay. Now, example for prokaryotic cell, bacteria. In bacteria, DNA is circular and we call that as nucleoid. Whereas in multicellular organisms, the DNA will be in thread-like form with so many genes and chromosomes embedded in it. That's the basic structural difference. Okay. I'm going a bit faster because all these are the very basic points which everyone know. Okay. Now, this is the point of interest where I discuss elaborately about the cell wall of prokaryote. I had taken the example of bacteria and I explained this in elaborate way. What are the different components involved in prokaryotic cell wall structure and how the cell wall becomes very rigid in case of bacteria. Okay. Now, see. When you take the prokaryotic cell wall, we are using cell wall, not cell membrane. Understand this word, cell wall. Cell wall you will find in plant cell and in prokaryotes. Remember that. That's the similarity between plant cell and prokaryote. Now, when you take prokaryote, example, I'm taking bacteria here. So, taking bacteria, I'm explaining so that you'll understand in a better way and I'll relate this structure to gram staining method. Okay. Now, when you take the cell of prokaryote, the cell wall is made up of peptidoglycans. Get the word peptido stands for peptides. Peptides are nothing but amino acids. peptides are bonded to glycons. Peptidoglycans. Glycon stands for carbohydrate. So what happens here? An amino acid is bonded to carbohydrate. How? Here, specific terminology you have to understand. N-acetylmurine, N-acetyl glycoside. Okay? N-acetylmurine, N-acetyl glycoside. See, take the structure. If under electron microscope, if you observe the cell wall structure of bacteria, this is how it looks like under electron microscope. The structural representation. Here the chain structures are there. See. Okay. And the chain structures are joined together by some dot-like structures in between. Then such chain structures are numerous and they are interlinked or cross-linked. You can use either words. They are cross-linked. See, the cross-linkage. Here comes the importance of any prokaryote. For example, bacteria. Assume that you got a bacterial infection. Suppose cholera, something. The basic example I'm taking. When you take cholera is caused by a bacteria, Vibrio cholerae. You know that. Now, how this bacteria can enter into human body and can survive and shows growth inside human body. It should be having some strong structure around which any of the enzymes or any of the immunity of human body cannot work. That's why infection happened. So there is the rigidity or there is a strengthened mechanical support given by outer surface of this bacteria, which is nothing but the cell wall of the bacteria. So the cell wall is made up of NAG, N-acetyl glucosamine, NAG, N-acetyl glucosamine. Here the words I given in elaborate way. N-acetyl glucosamine or glycoside, then NAM, N-acetylmurine. N-acetylmurine. When you take these chain structures, the chain structures are nothing but murine and glycoside. Murines are the amino acids, glycosides are the carbohydrates. And they are cross-linked. So this cross-linkage is very important. This cross-linkage is done by carbohydrates. So I'm drawing on the diagram. So these are the chain structures which are made up of amino acids and carbohydrates in the form of N-acetylmurine and acetylglycoside. And those structures of chains are cross-linked with each other. Very strong cross-linkage you can observe through carbohydrates. So this is how the structure of cell wall becomes very strengthened and gives mechanical support. It's very rigid and it will not be easily broken down by many of the enzymes of the infected host cell. So bacterial cell wall helps in its penetration. The word I'm using, penetration. Penetration nothing but in biology we call it as infection. Penetration into plant cell or animal cell. Bacteria can infect plant or animal. Okay, so this is how the cell wall in prokaryote, for example, bacteria, is made up of amino acids called peptides, carbohydrates called glycans. Basically, cell wall of prokaryote is made up of peptidoglycans, which gives rigidity to the cell wall and helps in infection. Explication about the prokaryotic cell. When you take eukaryotes, so just now I explained, eukaryotes, especially plant cells, only will have cell wall. Animal cells will not have cell wall. Now, plant cell wall is composed of cellulose. That's the basic component of plant cell wall. But other than cellulose, there are so many different components you will observe in plant cell wall. I'll explain all those points. Now, when you take the plant cell wall, the plant cell wall chemical composition I'll explain. Major portion you'll find water. Other than water, You will have some constituents like cellulose, hemicellulose, pectins, lipids, and proteins. I'll explain the whole cell wall structure and I'll let you know how the cellulose, hemicellulose, and pectins and remaining proteins, lipids are arranged. Okay, let's see that in the next slide. So here this slide explains about major portion of cell wall. That's the important point. That's why I given this slide. 30 to 60%. See, most of the cell wall contains water. Remember that point. Okay, remaining things you'll understand in the next slide. See, when you take the structure of a plant cell wall, the plant cell wall is not made up of a single layer, but it is made up of various layers. Okay, when you take the various layers of a plant cell wall, you will find different compositions. Plant cell wall, it is made up of basically 3 layers. Okay, the first layer is called middle lamellum. Second layer is called primary cell wall. Third layer is called secondary cell wall. I'm using the 2 degree, 1 degree to designate primary and secondary. Okay, so middle lamellum, primary cell wall, secondary cell wall. Assume that this is a plant cell. Just a cell inside a plant and take it. Okay. Now when you take a plant cell, it is surrounded by cell wall, we are saying. So if this is the cell wall, again the cell wall is made up of various layers. Now when you take the cell wall, the first foremost outer layer is called middle lamellum. Okay, then inside the middle lamellum, you will have the primary wall. I'm showing it with a different color. This is the primary wall. So this is the primary cell wall. Then inside the primary cell wall, you will have secondary cell wall like this. So this is secondary cell wall. Okay, so you will be having various components in the cell wall. The first foremost one is a middle lamellum. So when you take another cell like this is another plant cell. You will have communication between 2 plant cells through middle lamellum because the outermost layer is the middle lamellum. Understood? So here when you take the cell-cell communication happens through middle lamellum because that is the one which is the outermost layer. Okay. Then inside that primary cell wall, then secondary cell wall. Within that you will have the original cell. Now when you take the how primary cell wall and secondary cell wall are produced, there are different processes which are involved in production of primary cell wall and secondary cell wall. When you take the primary cell wall production, the process is called intersusception. Intersusception. Intersusception is the process where if you take this as a material and within this material, if above which you will have some growth like this, if this material is growing outwards, get the word outwards, outward growth. Then such outward growth is called intersusception. Okay, the word intersusception stands for outward growth. Outward growth of what? Already existing substance. Existing substance when gets expanded, that is called intersusception. The primary cell wall will be formed by this intersusception process. This is the process. Okay. So what does mean by intersusception? There will be some material already present and that material shows outward expansion so that the existing material gets thickened like this. So this is called intersusception. It's a process by which primary cell wall will be produced. Now when you take the primary cell wall contains peptides, cellulose, hemicellulose and some polysaccharides. And where you find the primary cell wall? Not in all plant cells. You will find primary cell wall especially in immature meristematic cells. Do you remember anything about meristematic cells? Anyone? Just give a blind definition of meristematic cell.
And the cell which are capable of division.
Yes, repeated division, repeated growth. Very good, Anna. Isn't it? We discussed meristematic cells, various forms, apical meristem, intercalary meristem, isn't it? So many lateral meristem, so many examples we have discussed, isn't it? So in all those areas, you will find immature meristematic cells, you will find primary cell wall. Okay, and how the primary cell wall is produced? By a process called intersusception. Intersusception. What does it mean by intersusception? There will be an existing substance. There will be substance present already, and it shows outward growth. It shows expansion. That is the intersusception process in which the cellulose, hemicellulose, peptides, all this gets deposited. And the thickening cell wall happens, which we call as primary cell wall. Not only in immature meristematic tissue, we'll find this primary cell wall. We'll find even in parenchyma tissue also. In parenchyma cells also, you will find primary cell wall.
And they are simple permanent tissue which fill up most of the spaces in plants.
They don't have intercellular spaces. They'll have numerous vacuoles. So all these characters we studied. Okay. So here our point of interest is how the primary cell wall is formed. Primary cell wall is formed by a process called intersusception, and we'll find primary cell wall with all peptides, cellulose, hemicellulose, and immature meristematic cells and parenchyma cells. They possess primary cell wall. Wherever you find these cells, you'll find primary cell wall. Okay. Then after primary cell wall, you'll have secondary cell wall. Now when you take the secondary cell wall, secondary cell wall is made by a process called accretion. Primary cell wall is produced by intersusception. That is the pattern of growth process of primary cell wall. Whereas secondary cell wall is formed by accretion method. What does mean by accretion? So, see, this picture explains the whole thing. When you take a plant cell, assume that this is the plant cell, they have taken the transverse section. Now, plant cell will have plasma membrane, above which you will have the cell wall. So, as of I told, the outermost one is the middle lamella, below which primary cell wall will be produced. So, see the violet or bluish violet one is the middle lamella, under which primary cell wall is produced by intersusception process. Means you will have some material, and upon that some more material will be added. So that forms the primary cell wall. Now coming to the secondary cell wall, see the bigger picture. So when you take the secondary cell wall is there between primary cell wall and plasma membrane, isn't it? This is the secondary cell wall layer, the marked one, the yellowish part. Now when you take secondary cell wall, the secondary cell wall is having very much cross-linked rigid structure. Now when you take the black color structures, the black color parallel lines, that is nothing but the cellulose. So cellulose is there in primary cell wall and even secondary cell wall. But there's a difference. When you take the secondary cell wall cellulose, there are more cellulose amount. And they are arranged in the form of fibers, not loosely arranged like in primary cell wall. Observe the difference. And when you take the green color structures, which are hemicellulose, there are less in primary cell wall, the green color one, pointing out in the diagram, see. But when you take the secondary cell wall, there are more hemicellulose. And you're having special structure called lignin. The oval shaped structures which are not there in primary cell wall. See, but there more in secondary cell wall. See, circling those structures. So those are called lignin. So when you take the secondary cell wall, in secondary cell wall you will have cellulose microfibrils, more in number, hemicellulose, more in amount, and lignin is the one which is exclusively present in secondary cell wall. So this process of various fibers which are interlinked and giving a rigid nature to secondary cell wall is called accretion. Accretion type of growth you will find in secondary cell wall. Accretion here, the word stands for numerous materials like the cellulose, hemicellulose and lignin are deposited, making the cell wall part very rigid. That's the growth of secondary cell wall happening through accretion method. Okay, get the words accretion and the one they have showed as a cellulose synthase complex, isn't it? The blue color one. Whenever you find the word synthase, it's something related to synthesis, producing by its own. Here we are talking about the cellulose fibrils, hemicellulose fibrils, isn't it? So the cellulose and hemicellulose fibrils will be produced by a complex called cellulose synthase complex, which you will find in plasma membrane. Observe the diagram carefully. It's there in plasma membrane, not in the cell wall. See, isn't it? So this is a complex which contains various enzymes required for the synthesis of cellulose and hemicellulose. Clear? So that this cellulose and hemicellulose gets deposited in primary cell wall and the secondary cell wall from the plasma membrane. See, that's why I explained in the starting of the concept about you will have a cell around which you will have cell wall. Okay, if it is a plant cell. Now around the cell, whether it is a plant or animal cell, you will have plasma membrane, isn't it? So when you take this as the plasma membrane, this is the plasma membrane. I'm showing on the slide. See, the green color bracket I'm keeping. That's the plasma membrane. So this plasma membrane contains a complex, cellulose synthase complex, which have the enzymes required for the synthesis of cellulose and hemicellulose. Now this synthesized cellulose and hemicellulose will be formed in primary cell wall. See, there is a connectivity, black connectivity, isn't it? Can you see that? So the black lines, the parallel lines are nothing but the cellulose. Here they have shown, see? I'm circling on the diagram. Can you see what I'm circling? Okay. Now the cellulose will be synthesized by this complex in the plasma membrane and it will be deposited there. So it forms the primary cell wall first. Then it will go secondary cell wall later. Then beyond that, middle lamella. This is the order. Middle lamella is the first one. Then primary. Okay, fine. So this is how we can say the methods of intersusception and accretion. What we are saying in these two ways, the primary cell wall and secondary cell wall will be produced. From where? Through this complex, which is having different enzymes in plasma membrane. Okay. Now, when you take the secondary cell wall, as of we said, primary cell wall will find in specifically meristematic cells and parenchyma cells, wherever they are present. Likewise, the secondary cell wall, if you take the secondary cell wall, you will find in cork. I hope you remember this word, cork, or wood. Both are different. Hardwood, I explained, hardwood. If you take the cut part of a trunk of a tree, you will have various layers around it, isn't it? The outermost hard structure is called bark or cork, isn't it? So this we call as hard structure, which will be in form of patches around the trunk of a tree. So that, and even the wood of a tree. which definitely will have secondary cell wall. So when you take the texture of cork or wood, they will be very tough, isn't it? And they will be very thick and rigid. So that explains they will have the secondary cell wall. So what we said, secondary cell wall is the thick rigid part of cell wall. So obviously you find this tough, thick, rigid part of secondary cell wall at the cork position and the wood position of the plants and trees. Now comes the question how this became very thick and very tough. Lignin. Lignin is nothing but an organic polymer. Lignin is not a protein. This is a different question they'll ask repeatedly. Lignin is not a protein. Even suberin is also not a protein. Lignin is a polymer, especially organic polymer. What are organic polymers? It is not a protein. It is an organic polymer. Polymer always stands for you will be having many molecules. Okay. So those many molecules will be cross-linked and forms a hard structure called lignin. And when you take suberin, suberin is also a polymer, but it is also made up of many molecules of fatty acids. Sterols generally we call. Okay, so all these are biochemical nature of lignin and suberin. Lignin and suberin, these are the special materials you will find only in secondary cell wall. Okay, you won't find in middle lamella, you won't find in primary cell wall, you won't find in plasma membrane. You will find these structures only in secondary cell wall. So lignin and suberin both are polymers, organic polymers, which are made up of many molecules. Generally, the question can be like this: how the primary cell wall is different from secondary cell wall. Then you have to go for these words: lignin, suberin, and all. Okay, where you find lignin, where you find suberin, you will find in the cell wall of trees, especially secondary cell wall part. Okay, I'll show a slide which explains the difference between primary and secondary cell wall. Here are the differences. When you take primary cell wall, you will find in all plants. Understand the word. But when you take secondary cell wall, you will find only in mature plants, or you can say trees. That's why we are talking about bark and cork and wood part. So those are the mature parts. When you take the position where you find primary cell wall, it is inner to middle lamella, isn't it? Inside middle lamella, you will have the primary cell wall. And when you take secondary cell wall is between primary cell wall and the plasma membrane. I have given the flow chart, isn't it? So between the primary cell wall and the plasma membrane, you will have the secondary cell wall. Then nature thinner one primary cell wall, whereas secondary cell wall is very thick and rigid. Understand the word rigid. Why it is very rigid? Because of the presence of lignin, suberin, and cutin. Cutin is also a polymer. These are all present only in secondary cell wall, whereas you won't find such additional materials in primary cell wall. Okay, in primary cell wall they're totally absent, whereas in secondary cell wall only you will find additional materials. The nature of growth is the most important point. Intersusceptional growth is for primary cell wall, accretional growth for secondary cell wall. And when you take amount of cellulose, it's low in primary cell wall. The previous slide you can observe the black color parallel lines. They are very less in primary cell wall, whereas they are very numerous in secondary cell wall. Means cellulose is high. And extensibility. Extensibility is nothing but the elastic nature, which we will find in primary cell wall only, but you won't find in secondary cell wall. Why? Why? Because you are having the rigid nature because of the lignin and suberin-like materials, the polymers. So they will not make the secondary cell wall elastic or extendable. It will be thick and rigid, only the secondary cell wall. Then arrangement of fibrils and hydration, these are the common points. You can observe in the previous slide. The loose arrangement of parallel lines of cellulose and all. But there is a parallel arrangement of cellulose and hemicellulose in secondary cell wall. So these are the differences between primary and secondary cell wall.